The CAMP-CRP complex is a positive regulator or activator in contrast to the repressor, and the lac operon is independently regulated both positively and negatively. Based on the information available, it may be proposed that glucose somehow inhibits phosphorylation of adenylate cyclase, thereby preventing CAMP formation. So अगर ग्लूकोज प्रेजेंट है तो ये CAMP सिंथेसिस को इनहिबिट करता है. The next challenge was to find the link between glucose metabolism and adenylate kinase phosphorylation. Studies indicated that the link is a glucose-specific phosphoenol pyruvate dependent phosphotransferase system or PTS. This system uses energy supplied by phosphoenol pyruvate or PEP to phosphorylate glucose as it transports the sugar across the inner cell membrane. This system requires 4 proteins. So this is the system, and here you can see the sugar is entered into the cell, and then this sugar is phosphorylated, and through glycolysis it is converted to PEP, and then PEP is converted to pyruvate, and then this is a protein which is enzyme 1. This is another protein, HPR, histidine containing protein, and then there are 2 other proteins, 2A and 2B. So these 4 proteins are required for this phosphorylation. 2 of these, that is enzyme 1 or E1 and histidine containing protein HPR, are also components of other sugar transporter systems and therefore are unlikely to be direct participants in a glucose specific phenomenon. The 2 other proteins, that is enzyme 2 or E2A and enzyme 2BC, are specific for the glucose transport system and are most likely participants in a glucose specific phenomena. So ये जो दूसरी प्रोटीन्स हैं यानी कि एंजाइम 2 और एंजाइम 2BC, मोस्ट प्रोबेबली ये 2 वो प्रोटीन्स हैं जो इस फिनोमिनन में पार्टिसिपेट करती हैं. E2A participates in the catabolite repression by 2 different mechanisms. The first mechanism is based on the fact that E2A can transfer a phosphoryl group from HPR, phosphorylated HPR or HPRP, to either enzyme 2BC or adenylate cyclase. The preferred substance is E2BC, which then transfers the phosphoryl group to glucose to form glucose 6-phosphate. However, when glucose is unavailable, E2BC will be fully phosphorylated and E2A phosphate has no other alternative but to transfer its phosphoryl group to adenylate cyclase. तो फिर इसका मतलब है कि E2AP के पास कोई ऑप्शन ही नहीं है, वो अल्टीमेटली फॉस्फोराइल ग्रुप को जो है वो एडिनाइलेट साइक्लेज पे ट्रांसफर करेगा. Phosphorylation changes the inactive dephosphorylated form of adenylate cyclase to the active and phosphorylated form of adenylate cyclase, which then converts ATP to CAMP, as you saw in last slides. Thus, glucose interferes with the conversion of inactive form of adenylate cyclase to the active form by phosphorylation of this enzyme. The second mechanism by which glucose influences lac apparent transcription is called inducer exclusion, which also involves the glucose transport system. When glucose is present, E2A phosphate transfers its phosphate group through E2B to the sugar, and the dephosphorylated form of E2A binds to lactose permease and inactivates it. So इसमें lactose permease जो है वो पहले inactivate हो रहा है. Lactose permease inactivation prevents glucose from entering the cell and being converted to allolactose. In the absence of allolactose, the repressor remains bound to the operator and lac operon is turned off. The CAMP modulation and inducer exclusion mechanisms may not be the only ones that contribute to glucose's ability to inhibit lac mRNA formation. तो सिर्फ ये दो मैकेनिज्म्स ही नहीं है, और भी कई मैकेनिज्म्स जो हैं इन्वॉल्व हो सकते हैं इसमें. फॉर एग्जांपल, रेगुलेशन ऑफ CRP synthesis may also be important in this regard.